Okay, so we're going to be talking about Punnett squares. What are they? Punnett squares are a tool that you can use to calculate the probability of organisms having certain traits. By the way, this is the very first vocabulary term on your portfolio, so you may want to go ahead and write this down. All right, so Punnett squares are the tool that we use to calculate the probability of inheriting certain traits. So probability is a fancy word of saying the chances. All right, so the reason why we use Punnett squares to get the chances is because each of these squares in a Punnett square represent a 25% chance, or like a 1/4 chance of that occurring. Okay, we're going to be talking about two different types of Punnett squares today. The first one we're going to be talking about are monohybrid Punnett squares, or monohybrid crosses. And this picture that we see here is an example of a monohybrid Punnett square cross, because it's involving one trait. So for example, this cross is dealing with one trait, or just one gene that codes for the colors of peas. And notice there are two alleles, two forms of the gene, a dominant one and a recessive one. All right, so one more time, definition of a monohybrid cross. It's a genetic cross that we use that involves one gene and one trait. And so when you're solving monohybrid crosses, each of your offspring are going to receive one allele from each parent. All right, so now let's learn how to solve them. We're going to start by watching this quick video, and then we're going to jump straight into practicing it for ourselves.
Hi, my name is Mary Poffenroth. I'm an adjunct professor of biology, and today we're going to have a little fun with Mendelian Punnett squares. Now, before we get started and jumping into our Punnett square problems, let's set down some ground rules. First, we're going to talk about alleles. Alleles are going to be like different flavors of genes. So, you go into the ice cream shop, there's 31 flavors of ice cream. Even though they're all different flavors, they're all still ice cream, right? Alleles are going to be those different flavors of genes that you have available to you. The next thing I want to talk about is dominant and recessive. In any relationship, some of those alleles are going to be dominant and some of those alleles are going to be recessive. Those dominant alleles, if they're present, they're going to have more of a say in what the outcome of that cross is going to be. And the recessives, well, they're going to have little to no say unless there are two recessives that are available. Also, in each of our crosses, we're going to have one allele from our mother and one allele from our father. Now, let's jump into our crosses. Today, we're going to do a 4-square Punnett square. So, you want to start out by just drawing a square. In that square, you're going to draw a line right down the middle from the top to the bottom and a line from left to right. Now, you have 4 individual squares. Each of those squares is going to be a probability, and each of those squares is 1/4, right? Or 25%. Next, we're going to put our parents' alleles on the Punnett square. Now, it doesn't really matter if you put male or female on the top, but generally people put male on top, female to the left. For our problem today, we're going to use eye color. The eye color alleles are going to be, for our father, it's going to be brown phenotypically expressed alleles. That means that that father has brown eyes, but his genotype, or the genes at play, his alleles are going to be big B, little b. That means that he's heterozygous, or he has 2 different alleles for that trait of eye color. It's a little important to note at this time that all of these letters are completely arbitrary. You can make up any letters you want. However, in any relationship, a capital letter or a big letter, like say big B, is going to be for the dominant. And a little letter, or a lowercase letter, in this case little b, is going to be for the recessive. Okay, so we have the father, big B, little b. He's heterozygous, meaning he has 2 different alleles. What about the mom? The mom has blue eyes. So phenotypically, her expression that you see on the outside is blue eyes. Her genotype, or the genes at play, are going to be little b, or homozygous recessive. Alrighty, it's time to do our cross. Now, we're going to take one from the top and one from the left, and we're going to bring them down into those squares. So, we have on our top, big B, little b. Then bring another big B, little b down. In our right square at the top, we have little b, And our bottom right square, we have little b, little b. Okay, what does that mean for the probability of getting an offspring with what kind of eye color? Now remember, each of those squares is going to be 25%. So we have a 25 plus 25 or 50% chance of getting an offspring with brown eyes. And we have a 25 plus 25 or 50% chance of getting an offspring with blue eyes. And for a fun little extra problem when you're sitting around the dinner table, take the eye colors or the phenotypes of your parents. Even though you won't know the exact genotypes or the genetic makeup behind what you're seeing on the outside, do some crosses and see if you can find out the probability that you could have actually had a different eye color.
Okay, not bad. Okie dokie. Okay, so here's question number 1. It's a monohybrid cross, and here's what it says. It says brown eyes are dominant over blue eyes. If 2 hybrid brown-eyed people married, what are the chances that they will have a blue-eyed child? All right, so whenever you're solving a Punnett square, there are certain steps I always want you to follow. You can find those steps up. You scroll up a little bit. There are the 4 steps here for how to solve a genetic problem. The very first thing you're always going to want to do is make a key. All right, now I made it really easy for you guys because I already have the key set up for you. In your key, you're going to write which trait is dominant, which is always going to have an uppercase letter. And you're going to write which trait is recessive, which is always going to have a lowercase letter. All right, so that is always your first step when you do a Punnett square problem. Even if I do not give a key or a space for the key for you, you have to know on a separate piece of paper, that is the very first thing you should always draw out or write out. What the key is, what each of your letters are going to be, and what they're going to represent. Okay? Next thing to keep in mind is you have to figure out what the genotypes of the parents are going to be. So what letters will your parents have? That is very, very important. You got to figure out what you're crossing. Here's a couple of hints for you. If you read a question and it says that the parents are pure for a trait, pure is code word for homozygous. Remember, you can be homozygous dominant and have 2 uppercase letters, or you can be homozygous recessive and have 2 lowercase letters. All right? Or if you're reading a question that says the parents are hybrid, like the one we're looking at for number 1, or if they're mixed or they're carriers, that is code word for heterozygous. Remember, heterozygous individuals have both an uppercase letter and a lowercase letter because they have both the dominant allele and the recessive allele. All right, so once you've set that up, once you've made your key, once you've correctly identified the genotypes of the parents, now or then you can do your Punnett square. And the very last thing you'll do is answer the questions. All right, so that's going to be the order I'm going to use to solve question number 1. Okay, so the very first thing I need to do is fill out my key. So it tells me in the question that we have 2 traits, brown eyes and blue eyes, and it made it very easy. It told me which one is dominant. So it says brown eyes are dominant. So is that going to be my uppercase B or my lowercase b? Uppercase B is going to be brown. All right, and that must mean blue eyes is recessive, so that's going to be my lowercase b. Hopefully you guys online, I'm giving you the answers to question number 1. This is question number 1 in the portfolio, so you can just be typing or writing this along with me. All right, so that's step 1. We've made our key. Yay! Step 2, figure out our cross. All right, so it said that the parents are 2 hybrid and they have brown eyes. So remember what I said before? Hybrid is code word for heterozygous. So heterozygous individuals are going to have 1 dominant allele and 1 recessive allele. They're going to have 1 big letter and 1 little letter. So if both of my parents are heterozygous, that must mean they have 1 big B and 1 little b. 1 dominant allele and 1 recessive allele. And both of them have a dominant allele and a recessive allele. All right, does that make sense so far? This is actually the most important part. Making your key and setting up your parents' cross. Because if this is wrong, everything else will be wrong. So this is the part you want to make sure you're doing right, this setup. Okay, now what's the fun part? We get to fill in our Punnett squares. All right, so to fill in the Punnett squares, you're going to take the alleles from one parent and put them on the top, and the alleles from another parent and put them on the side. It doesn't matter which one you put where. It really doesn't matter.
You can help go ahead and fill in the diagram so it looks like a square. That helps you.
Oh yeah, because the way it printed on it had like colors on the top, but the way it printed was weird. All right, so here we go. I'm gonna put this on the alleles for this period here. I'm just gonna put it on the top, because. So I'm gonna put a big B and then a little b. All right. Now I'm gonna take the alleles from this parent and put them on the left-hand side. So once again, they had a big B and they had a little b. Are you still with me? All right. Okay, here we go. Now we're going to fill in our Punnett square. To fill in a Punnett square, you're going to drag and drop. You're going to drag over the allele from the left, and you're going to drop down the allele up top. All right, so I'm going to drag and drop. Big B, big B. I dragged over, and I dropped down. All right, I'm going to do the same thing for this box. I'm going to drag over this little b, and I'm going to drop down this big B. All right, now pause. I did something. I wrote the capital letter before the lowercase letter because I think it looks better that way. You could write it this way, and it would be fine. But it looks weird. So everyone typically writes the dominant allele, then the recessive allele, like that. It's appearances.
But if you wrote it that other way, it means the same thing.
It means the exact same thing. So if you wrote it that way, I wouldn't be like, wrong. No. It's still heterozygous. Cool.
It's not as pleasing to me.
Yeah, it's just not as pleasing. So no one does it. Like, no one ever does it. All right. So here we go. Let's do this box. We're going to drag over this big B. And I'm going to drop down this little b. Whoop. And then last one, I'm going to drag over this little b. And then I'm going to drop down this little b. All right, Punnett square done. Questions on how I filled out this Punnett square? My friends online, were you able to do this in your portfolio? Yay or nay? If you're online, can you type in the chat and say you got it? Just so I know. Good. All right, well, 5 is better than none. Well, take it and go. All right, beautiful. Okay, so now the next question on the next section asks about the probability. So remember, each of the squares in a Punnett square represent 1/4. I'm going to draw another one here. You guys can see. Actually, am I on camera anymore? I can't even do it. Okay, good. So each of these boxes represents 1/4 chance. Remember, another way of writing 1/4 is 25%, like a quarter. Right, so each of these boxes is 1/4 or a 25% chance. So keep that in mind as we answer some questions about the probabilities. Remember, probability is just a fancy word of saying, what are the chances of something happening? All right, so the first one asks, what is the probability of having an offspring with 2 uppercase B's? Now, there's a fancy word for this, 2 uppercase B's. So it's the same alleles, so it's homozygous. But they're 2 uppercase letters, so they'd be dominant. So you would call this genotype homozygous dominant. All right, so What are the chances that they're going to have an offspring that is homozygous dominant for this trait? So if I look at my Punnett square, how many boxes has two uppercase B's? One box. So what is my chance if it's just one box with that genotype? 25%, aka 1/4. So you could write either one. You could say 1/4, or if you like percentages, you could say 25%. Either one works. All right. To the next one. The next one is asking us for the probability of having an offspring with the genotype 2 little b's. The term for 2 little letters is homozygous. Remember, homozygous means same. But they're lowercase letters, so it's homozygous recessive. All right. So looking at your Punnett square, what is the chances of them having an offspring that is homozygous recessive? 2 little b's. How many of those boxes do you see in your Punnett square? 1. Exactly. So it's just this box here. So that's one box. One box equals 25% or 1/4. All right. So once again, 1/4, aka 25%. All right. Next, it's asking about an offspring that has a big B, little b. This genotype is heterozygous. Remember, hetero means different. All right, so what are the chances that they'll have an offspring that is heterozygous? 50%. Why 50%, Tyler? Exactly. There's 2. 1 and 2. So each of those boxes represents 25%. 25 plus 25 is 50. Think of quarters. That's the easy way to think of it. Each of these is a 25-cent coin, a quarter. If you have 2 quarters, you have 50 cents. A Russian. No, just kidding. You can also say 1/2. That's the same thing. That's right. 1/2 or 50%. All right. So these probabilities so far, these would be genotypes. That term genotypes describes the genes that an offspring can have. All right. Now, these last 2 down here, these are asking questions about the phenotype. Remember, phenotypes means physical appearance. So, what are the chances that they're going to have an offspring with brown eyes? 75%. How? Okay, well, let's look. Brown is my dominant allele. So any of these boxes that has that dominant allele is going to have the dominant trait. So this box, oop, got it. It's going to be brown eyes. This box, whoop, all you need is one. All you need is one dominant allele. It completely masks the recessive allele. So this one, brown eyes. This one, got it, brown eyes. That's 3 out of 4 boxes. And another way of saying 3/4 is 75%. Think quarters. If you have 3 quarters, you have 75 cents. All right. Then last question. What are the chances that they'll have a child with blue eyes? %. Just one box remaining. Right? So this box has 2 of the recessive alleles, which means they'll have the recessive trait of blue eyes. That's 1/4, aka 25%. Beginning of the portfolio, a dihybrid cross. So a dihybrid cross is a genetic test that instead of involving one trait like a monohybrid cross, it's going to involve 2. All right? So we're not just going to be dealing with 1 letters, not just B's. We're going to do B's and C's. We'll do fur color and eye color, height and shape, texture, and size. 2 traits. All right? So in these types of crosses, parents aren't just going to give 1 letter. They're going to give 2 letters to each of their offspring, 2 alleles to each of their offspring.
Number 8.
We're about to get to number 8. We're moving, or I'm kind of jumping around. I just want to get all the information out, then you have the rest of the class to work through it. All right? So here's how a dihybrid cross looks like on a Punnett square. Much bigger, right? Much bigger. Instead of having just 4 boxes, you're going to have 16. Here's the good news. I don't want you guys to make a dihybrid Punnett square. Yay! You don't have to do that. I do need you to know how to set up the parents' gametes on the tops and the side. So what you will need to know is the possible gamete combinations, the allele combinations of the parents. That's all I need you to be able to do for dihybrid crosses. You won't have to fill in a Punnett square. Just know what would go on the top and the side of the Punnett square. Because those would be your gamete combinations. Now, what is one thing you notice about the top and the side of the Punnett square that is different? How many letters is each parent giving? 2. All right, it's 2. So there's a little trick that you can use to figure out which letters each parents are going to give. And it's called foiling, like math. First, outer, inner, last. And so you're going to use this foiling method to figure out the gamete combinations of the parents. All right? So I'm going to show you with the example on the screen, and then we'll do the one in the portfolio. Readjust myself. Okay. All right, so remember, in a dihybrid, all right, so the last thing was the question cross. You're dealing with 2 traits. So each parent is going to give 2 letters. All right, so for this example, we're just going to say trait number 1. I don't care what it is. And trait number 2. Right? Trait number 1, we're going to use the letter A. Big A for dominant trait number 1. Little a for recessive trait number 1. Trait number 1 could be anything. Hair color, eye color, height, I don't care. Trait number 2, we're going to say B. So big B, little b. Cool? Doesn't matter what it is. So let's pretend we have a parent who is hybrid for both traits. If he's hybrid for both traits, that means this parent is going to have a big A. Oh, Emerson, I have an electric one over there. Don't use that on the screen. Over there by like the wardrobe. On the file cabinet, yeah. They're going to have a big A and a little a, and a big B and a little b. 2 traits, dihybrid. All right? So now you need to figure out what are the gamete combinations. And to do this, we're going to FOIL. F standing for first, O standing for outer, I for inner, L for last. All right, so here we go. Starting with the first letter, A, I'm going to combine it with the first letter over here, B. The first letter. So that first combo would be A and B, uppercase A, uppercase B. Next, we're going to do outer. So I'm going to stick with my first letter, and I'm going to combine it with the one on the outside. So that's capital A, lowercase b. All right. Next, inner. So I'm going to do my second A and my second or my inner B. which is a lowercase a, uppercase B. And then last, second letter, last letter. All right, I sing a song to help me do this. This is how I do it. I'm just going to write it again with my song. I go first letter inner, first letter outer, second letter inner, second letter outer. And I always get it right. First letter inner, first letter outer, second letter inner, second letter outer. That's how you form. And that's it. That's all you need to know how to do for a dihybrid cross. Tell me the gamete combinations from those parents. All right, so let's practice. Let's go to question number 8 in the portfolio. Question number 8. Notice that all I'm asking you to do for question number 8 is to make a key and tell me the gamete combinations for the parents. That's it. We're just going to practice foiling. So you find question number 8, pull it up on the screen. 6, 7, 8. Okay, here we go. Okay, so I'm going to make my key. I'm going to make my key. My key is going to be a little bit bigger, though, because now I'm dealing with 2 traits. 2 traits. All right, so my first trait I have are trotters and pacers. So I'm going to use capital T and lowercase t. It says that trotters are dominant over pacers. So I'm going to make my capital T trotters and my lowercase t pacers. Number 8, but the one that you have the research version. So your number is not the same as theirs. You're welcome. All right, so pacers. Okay. And then fur color, there are 2 types. We're going to use big B and little b again. I like B's. And it says black is dominant over chestnut. So black is going to be our uppercase letter, and chestnut is going to be our lowercase letter. Are you with me so far for how I made that key? It said trotters were dominant over pacers, and that black was dominant over chestnut. Beautiful. All right, next, we need to figure out what we're crossing. So it says, what are the chances of getting a chestnut pacer from a cross between 2 hybrid, oh, that word again, 2 hybrid black trotters. All right. What is hybrid code word for? Thank you, Greer. Heterozygous. So if they're hybrid for both traits, that means they're going to have a big and little letter of both letters. So they'll have a big T and a little t, a big B and a little b. All right. Now, in terms of finding the gamete combinations, this is easy because the parents have the exact same genotype. So you only have to do it once, right? So I'm gonna take one down here, or down here, and we're going to FOIL. Remember, FOIL, first, outer, inner, last, and I'm gonna sing my song as I do it because the song is what works for me. All right, here we go. First letter inner, that's capital T, capital B. First letter outer, that's capital T, lowercase b. Second letter inner, lowercase t, uppercase b. and second letter outer. Lowercase t, lowercase b. Done. The end. That's all you need to be able to do for a dihybrid cross. These are the Gammy combinations.